So the interpretation for all of these is also found in your lab manual, so you don't catch what I say up here. What you're going to do is remember you set up 3 fermentation tubes per organism. So you set up 1 for glucose, 1 for lactose, 1 for mannitol. What you're going to do is look and see first, is there a color change? What color did your tube start out as? Do you remember? Red. It was like the orangey red, phenol red at neutral pH. Your tubes may not be red anymore. So you're going to notice, is there a color change? If it's red, that means it's negative for acid production. If it's yellow, it's positive for acid production, meaning it does ferment the sugar source in the tube. Then you're going to look at the Durham tube. Remember, the Durham tube is that upside down tube in your fermentation tube. You're going to look to see if there's a bubble. If there's a bubble, and all that means is the Durham tube is no longer full. Then that's positive for gas, meaning as a consequence of fermentation, the bacteria produced gas. If there's no bubble, then that's negative for gas production. And usually you'll see possibilities. There are only 3 possibilities. Negative for acid, negative for gas, positive for acid, positive for gas, or positive for acid, negative for gas. You'll never see negative for acid, positive for gas, unless something went wrong with contamination. So, I'm going to have a look at mine. I'm going to show you some examples. So, I have my glucose E. coli tube. The media has turned yellow, so that's positive for acid production. The Durham tube is no longer full to the top. I can see some air trapped in there. Right there. There's a bubble. Yellow bubbles. So I'm going to call that positive for acid production, positive for gas production. This is an example of positive for acid, positive for gas. Positive for acid, positive for gas. You're going to want your sterilizer up to high, not low. Yeah, so make sure the red light is on, not the green one. We want to kill the bacteria. We don't want to give them a spa treatment. Okay, so now I'm looking for. So let's see what we have here. This is also positive for acid, positive for gas. You can see there's a lot of gas in that Durham tube. And even though it's not completely yellow, some of it's still a little red at the top. If there's any yellow in there, call it yellow. So start having a look at your tubes. I'm looking for a negative example to show you. Here's a negative example. This is my Proteus vulgaris. The media is red. We'll go over today why it's hot pink instead of orangey red, unrelated to that fermentation, but the Durham tube is completely full. So that would be a negative result. So you're going to look at your tubes and record your results, positive for acid, negative for gas, negative for acid, positive for gas, up on the board. Since we have duplicates, if the group before you gets there first and your results don't agree with that group, then put yours underneath. Otherwise, you can just put a check mark next to it.
That one looks like maybe
And remember,
it got
your
something in it.
tubes may be in someone else's
Yeah.
drawers. Yeah, of course. And you're going to do this for glucose, lactose, and mannitol.
Let me see it taken picture. I think all of these look like they're negative.
Yeah. But they're not yellow, right?
Just count the whole thing.
Yeah, so negative for
Yeah.
acid.
Acids, but 1 for.
Yeah. Well, they shouldn't be. I think that the tiny bubble we got in one of them is an accident because
It's acid?
yeah, because hers had a lot more gas in it when it was
Yeah.
positive and
If you're
also
not sure, call me over and I will help you interpret.
You want me to ask her?
sure.
Professor?
So, this is a good example. This is E. coli, right? How did I know? Sometimes there's so much fermentation going on that it loses all the phenol red gets used up and you get this white color. This is like positive for acid, positive for gas. And that is something that I like really well. Yeah.
You saw negative bubble? It's a.
No. I would say that might be when you stirred it, you introduced a little air in there. It's not significant. But what is that, produce vulgaris?
Yeah.
What part?
Glucose and mannitol. This one is mannitol.
Yeah. Make sure you record it up there. No gas. Remember to work with your partner. Make sure your partner agrees with your interpretation. Devin, why don't you choose? Pull them out and start looking.
I see them right.
Who has Proteus vulgaris?
Me.
Who's me? Okay. Can I see your glucose?
This is brown.
Acid is first. Good question. Yeah, last negative. Yeah, no, that one's good, but your first one. See how your media is still crystal clear? That's how you know it didn't grow.
Oh.
So if something doesn't grow, then it's not going to be yeah. So you're really kind of just in a broken spot. So when you evaluate, just look to make sure the media is out to get to the part. But otherwise, yeah. So that's why that describes it. Okay, start recording. bubble, so it's positive for gas. Yeah, if it's already up there, just check it out. But mannitol, yeah. It's yellow. It's yellow. It's yellowish. It just isn't. Oh, it's very positive for gas. See how big that bubble is? It's not full anymore. So you can see the end of the bubble kind of leaves the water line. Yeah, it's yellow. There's a little yellow, call it yellow. That's definitely positive for acid. It's yellow.
Positive for acid on red, red is negative, so
Red is negative. It's not red, so it's positive.
negative positive.
This is a different test. This is not on. Right now, we're just doing these. We have to add something to that one.
Okay, so it'll be positive positive.
You can't, that's not that test. You can't say anything about that test.
Okay,
Yeah, we're just looking at the ones with the derm tubes.
and that's not positive.
Okay. Just gonna look and make sure my results agree.
And that's not good.
E. coli is correct. E. aerogenes is correct.
Oh, E. coli.
E. coli should be positive for everything, as should E. aerogenes. Proteus vulgaris should be only positive for glucose, and it is. I love it when things work out theoretically. And Pseudomonas aeruginosa was giving me trouble yesterday, and it's still giving us trouble. That's not theoretical, but we have to go with the data we have, and my data agrees with it. Glucose. But two positive negatives. So we'll have another look at that. Lactose, I have negative, which agrees. And mannitol, I have positive, negative. So we'll go with that. And just make sure you record these, because this is what you're going to compare to for your.
So, Professor, even this is our control group that we're doing experiment, but we only use the result from the theory one, not the
No, we don't use theoretical results. We only use our actual results.
actual. Okay.
Which are these.
Yeah.
So right now, you each set up one organism. You're going to use the table of all your classmates' data combined to figure out which one your unknown is in 2 weeks. So you're going to get a you're going to set up all these tests independently using your lab method.
Oh yeah.
So this is really important.
So if
So can I finish? Yeah. So this is really important. You're going to set up all these tests. The next week, you are going to interpret them. When you interpret them, this is the chart you're going to compare it to. We're making one this week for carbohydrates, one next week for protein. Not next week. We're not going to be here next week because it's spring break. Have a very happy spring break. The week after, we're going to analyze our protein metabolism results and make a similar parts to interpret your data. One of the first parts of the unknown is going to be look at your fermentation tubes. Are there any species you can eliminate as your unknown based on your fermentation tube results? Let's say your fermentation tube say positive. What can you eliminate? Yep. So you're still between E. coli and aerogenes. So if you got negative, positive, negative, then you could eliminate 3 of them, and you would know you might have P. vulgaris. Of course, you're going to have to go through some confirmatory testing for that, because one test can be wrong. Many tests, you're looking for evidence to support your claim of what your unknown is, and that's how it'll go. The template is up on Brightspace for the unknown report. If you want to sneak ahead and have a look at it, you're more than welcome to. You just can't write anything until you get your unknown results. But it's okay to familiarize yourself with it. Kimmy had a question?
No. No, okay.
Did I answer your question or you forgot what it was?
Yeah, because the one I wanted to ask is like one of my tubes I didn't grow at all. So
Yeah, so you use the data. I have
Yeah.
backups for everyone.
Yeah, so that's why
You're not
when-
going
yeah.
with your data, you're going with what's up
Yeah,
here.
so that's-
Because that's the whole thing. Listen, any one of us could make a mistake or have something not grow. It's okay. That's why everyone has a backup, plus then I set up all. I'm doing it again today. I'm setting up everything as a backup, just in case maybe your loop's too hot, maybe you didn't get enough bacteria on there. If it doesn't grow, I've got it. Don't worry. So make sure you're writing these down in your chart when we're done. If you want to take a picture of this, it's fine. Just make sure you have it. Okay, so now we're going to move on to the OF glucose. What does OF stand for?
something fermentation.
Something fermentation. So what's the O?
Oxidative.
Oxidative fermentative glucose. So we're trying to determine if glucose is used oxidatively or fermentatively. And again, I have misplaced
Is
my
that involved that you don't know on first?
I know. Oh, there it is. What?
Is that involved with step 3 and first cycle?
It involves not step 3 of the Krebs cycle, but the whole Krebs cycle. So if something uses glucose fermentatively, that means it is using glycolysis over and over and over and over. So it's doing only substrate level phosphorylation. Remember, fermentation regenerates NAD+, so glycolysis can continue. But some bacteria can use glucose oxidatively through respiration. Respiration, in this case aerobic respiration, involves glycolysis, followed by the Krebs step, followed by the Krebs cycle, to make electron carriers, NADH and FADH2, for the electron transport chain. We'll be talking about that a whole bunch today as we go through some of these tests as well. But for 38. 38. So as far as this next test goes, OF glucose, we're looking to see if the bacteria use glucose oxidatively through aerobic respiration only, or if they're capable of using it fermentatively only. So we set up two tubes, one with oil, one without oil. What was the purpose of the oil?
So they cannot breathe.
Stop the see which is bacteria.
What's it to keep out? Oxygen, yeah. You don't want oxygen on there. If you prevent oxygen, oxygen is the final electron acceptor in aerobic respiration. If there's no oxygen, there's no aerobic respiration. We don't really use the term breathe, because bacteria don't breathe, but so we're looking to see if it's fermentative use of glucose or oxidative. So we have the oil and the no oil tube.
I have a question.
Yeah,
So the oil is to force the bacteria into
it's not to force the bacteria to do anything. It's just literally to provide a layer to keep out oxygen. That's all it's doing. So if there's oil on top, there's no diffusion of oxygen into the media from the air. So that was something in the AI. A lot of you were saying, oh, there's no aeration. It doesn't matter if there's aeration or not. Aeration is just like constant mixing. It's just the act, like your tubes right now, if you look at your media, there's an interface between oxygen and the tube. There's some diffusion happening where there's oxygen at the top of the tube and not oxygen at the bottom of the tube, because gradients naturally happening in your Winogradsky columns too right now. So just keep that in mind. Aerating means we're constantly mixing, so there's oxygen at the bottom as well as oxygen at the top. But the purpose of the oil is to keep out the oxygen. So, what was the pH indicator in this test? Bromothymol blue, correct. So, bromothymol blue turns what color in the presence of acid? Yellow. So it was green to start, remember. So if both your tubes, the oil and the no oil, turn yellow, fermentative, meaning the bacteria is using glucose fermentatively. Because remember, fermentation is an anaerobic process. It's not anaerobic respiration, but it is an anaerobic process, meaning it doesn't require air. It's not harmed in the presence of air. So what this is telling us is that the bacteria can use glucose in the presence and absence of air, meaning they're using fermentation. If you look at these tubes with the oil and the no oil, and you only see yellow in the no oil, that means this bacteria uses glucose oxidatively because it requires oxygen to use glucose. So that is an oxidative result. So you're going to look at both your oil and your no oil tubes simultaneously. I have two tubes here for E. coli, one with oil, one without oil.
Can you hold it?
They are both yellow, so what is my result? Fermentative. If one of them, if the no oil was green, if the oil was green
Take the
and the no
name,
oil
so
was yellow, what
the
would that
label.
be? The other one, yes, oxidative.
Okay, thank you.
So have a look at both your tubes, and then before you we'll see how this goes, because yesterday's was. Yeah, just put a check if it's green.
Did y'all get this in the book?
Okay. Can we distinguish any of these bacteria by this test? No. We should have been able to. So Pseudomonas aeruginosa should be oxidative use of glucose only. However, it didn't work for my class yesterday either. I don't know why. Everything else tested correctly for P. aeruginosa, except for the fermentation tubes. The MR and BP and the citrate worked out okay. So when you write your notes, this might not be an informative test for your organism, but you do it anyway. If it does turn out working and some of you get oxidative, then you will have support in theory for it being Pseudomonas aeruginosa, but you will still mention it in your report. But our controls did not work out that way, so we still have to proceed with other tests to confirm. So as long as you can explain it, it's fine.
Sorry,
Yeah.
repeat again which one is oxidative instead of fermentation?
So Pseudomonas aeruginosa should be oxidative.
Nosa, the last one?
Yeah, Pseudomonas aeruginosa should be oxidative. However, it is not. So we can't pretend it is. In the slides, the lab review slides for this week, there are theoretical data. Don't use that theoretical data to write your report unless I tell you it's okay, meaning when we set this up for your unknowns, all of a sudden it decides to work. Then we'll talk about it. But for now, you're using this data, not the theoretical data on the slides, okay? So be careful when you write your report, because sometimes students will write their report based on that theoretical data, and your report will be wrong. You won't come to the correct conclusion, because that's not what happened with our controls. So the whole idea is you're comparing to the controls and making a process of elimination to determine what your unknown is. It's pretty straightforward. So the next test we're going to look at is the methyl red test. And I have to think through this. So methyl red, thank you, is a test for mixed acid fermentation. So mixed acid fermentation is when the carbon source is used and produces not only just lactic acid, but lactic, succinic, formic, large drop in pH. Phenol red turns yellow at what pH? 6.8, right, very sensitive. Methyl red doesn't turn red. It starts out as yellow at neutral pH, and then it turns red at pH 4.4. So it's detecting a much larger drop in pH. It's less sensitive. So we can determine a little drop from a big drop. Phenol red can't do that. So for this test, what you're going to do, I'm just looking for my methyl red, there it is. So you're going to use your methyl red tube, which you marked MR last week. So I have 4 of them. And you're going to take a bottle of the methyl red pH indicator. And I'm just going to remind myself, I believe it's 10 drops, 5 to 10 drops. But I want to make sure, so I'm going to look at my manual. 5 to 10 drops. So I have methyl red here. You're noticing it's red. The red only happens at an acidic pH. We hold it at an acidic pH for stability. So it's red now, it's fine. So I'm going to show you what happens. I'm going to add it to my E. coli tube. I can see it grew nice down there. So it is capable of mixed acid fermentation. There should be acids in there. So what I'm going to do,
That'll be 4 and after.
I'm going to just add 5 to 10 drops of methyl red and see what happens. I added 10 drops, quick mix. It's red. So that would be positive for mixed acid fermentation. Now you might be saying, well, you added something red to something yellow. Yeah, it's going to stay red. I like it. Question everything. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That's science, so it's fun. Yeah. So add your methyl red, give it a quick shake, see if it's red or yellow. If it's red, it returns red, so you're adding 5 to 10 drops of methyl red. If it's red, positive for mixed acid fermentation. If it's yellow, it's negative. Make sure you're grabbing the methyl red. Have a couple of tubes, but you'll have to share and pass it on. They're up here, and I have one. I'm just going to finish my controls, and then I'll give it back. Does anyone need methyl red?
You know,
Let me see. You know what? Wait for someone else.
I don't know how much.
Yeah.
It's orange. Yeah, it's hard to tell if it's.
Those are your perfect textbook results. So in that we can see that. Equal Harris and equal iron holds positive for mixed acid fermentation. Urogenes, keruginosa are both negative. Make sure you put the methyl red back. So, positive, negative. Next, does take a little while. Take 15 minutes to incubate. Yeah.
You have to, the prep side, you have to remember the processes as well, serve something.
No, I want you to remember just the inputs and the outputs. So, what goes in? Okay. You need 3 NADHs, 1 FAC, 1 ECT via substrate level phosphorylase each time it's
And last is absolute citation, that's what you
oxaloacetate. Yeah, but I'm not asking you to remember. So if the lecture material is confusing, you can always email me about it. But in the video library, there is a video of me with a notepad going through the entire process in a lecture format. It's only like a 5 or 6 minute as well. So that is what I want you to do. I don't need you to memorize every step of the process. I don't want you to memorize every step of the process. If you want to do that on your own, you can skip it. But you don't have to for the purposes. For biochemistry class, yeah, they're going to make you memorize it. Maybe for one of your previous biology classes, you have professors asking you to memorize it. If you know it, great. It's not going to hurt you. But for this class, the inputs and the outputs are sufficient. And knowing what happens to those electron carriers next to one. So, methyl red, report positive, negative, positive. And this the both pro-scour test is next. What type of fermentation are we testing for with both pro-scour? Remember last week? Alcoholic fermentation, right? The test for alcoholic fermentation. Okay. So you start in this tube with glucose. And you ferment it. Meaning, you produce pyruvate. Pyruvate is then going to get reduced to acetone. NH+. Acetone is an intermediate before 2,3-butanediol, which is an alcohol, and ethanol. The Vogt's pro-scour test detects acetone. We're going to do this by adding 2 reagents, mixing it a little bit, and waiting 15 minutes. I'm just checking to see how many drops we need to add. 18 drops of each. So you are going to add 18 drops of Barrett's reagent A and 18 drops of Barrett's B. And then you're going to wait 15 minutes. If it's pink, it's positive, meaning it does use alcohol alcoholic fermentation. If it stays yellow, it's negative, meaning acetone is not present and there's no alcoholic fermentation going on. So when you come up here, you don't need the methyl red anymore. You're going to take 1 container of Barrett's A and 1 container of Barrett's B. Make sure you read those labels. And very simply, you're going to add 18 drops of each into each of your tubes. I want to show you what it looks like, just because it does look a little different.
I smell.
Yeah,
So
doesn't smell great. Don't worry about it. Now I've added my A to both tubes. Now I'm going to add my Barrett's B. If I have more than 1 tube, you only have 1 tube, so you just do it once. It's going to look a little thick and milky. That's normal. Just give it a quick mix. Let it sit for 15 minutes. I'm going to do the same with my second tube. Mix. Let it sit right now. It's almost kind of identical. Okay, yeah. You gotta time it for 15 minutes. So when you're ready, you can go get a set of Barrett's. I wanna work out. To be honest, that's the most exercise I get some days. Remember, you have to take pictures of your photographs. Now would be the perfect time to do that.
you want to do that? I think you do it better than mine. If you need, we see anything happen. But she wanted to against the sunlight.
Usually I feel like this is better.
Yeah, but like both sides.
Okay. You only start with the other side for some glow.
Okay, so you can
But
put
we have-
whatever works for you.
Is that?
Whatever works for you. Hold it or anything. Yeah, set up the time.
Yes. Into your BP tube, you're adding 18 drops of A and 18 drops of B. Give it a quick mix and let it sit for 15.
Let's do the same, right? Only the water.
Yeah, the water is getting down.
Yeah.
18 drop.
And then.
This for now.
Professor, this one ran out.
Okay, put it to the side next to it and get a different one, 'cause I think there's another empty one there.
But she had to use, swirl it around, so that this is all, just,
I still have about 9 minutes.
What time is it? 15 minutes?
yeah,
10:01.
start timer.
10:01.
Like I said, we should be done a little early today because if you missed it when you came in, you know maybe. Fortunately, the strains we need to set up our bioremediation experiments did not grow sufficiently to start the experiment this week. Like you and I, they all need a nice spring break to rest and grow and recover. So we will be setting up the bioremediation report next week, not next week, when we come back from spring break during that lab, and your due date will be pushed back. Part of we don't know. Hopefully we get everybody together. Good. which may lead to anaerobic fermentation.
Thank you. Yeah, that one was weird. I don't know how I didn't notice that last week.
Yeah.
do an internship. You should check that. Yeah, they put that. But that probably has to get February. I was a very, very good internship in China, but I couldn't because me and my boyfriend were kind of very busy at the end. Exactly the day we started, I made an internship there, so I didn't apply at all.
Somehow look at my computer right now. E. coli.
How is it? 16?
Yeah, it's me. Good. Did you have it as well?
Yeah, the ring, the red ring. A little bit.
It's just the yes. Okay. The last test, so how many minutes do you all have left?
5.
5?
Professor,
Yeah.
I have a question.
Yeah, of course you can ask me a question.
So, what made you stay in academia instead of industrial?
My first reaction is I didn't really, I guess I am in academia. But my role here is primarily teaching, and I really do love teaching. My parents were both teachers. If you went to IS 96 or IS 88 in Brooklyn, you might have, well, and you're old enough, none of you are old enough, you might have had my parents for either. My dad was a choral music teacher, he sang, he played piano. My mother was an Italian, Spanish, and English as a second language teacher in junior high schools for over 30-something years. They passed that love of teaching on to me, and it was always my favorite part of any job was teaching. I was never really in industry, I was in government. So I've been a lifelong civil servant. I enjoy the service aspect of it. Bless you. But I started out my career working at the New York State Police as a lab technician, and I went back to grad school. I went to NYU School of Medicine in Manhattan, where I was in the microbiology department there, and it was great. I couldn't find a job in microbiology after my PhD. I didn't want to do a traditional postdoc, which is where you do more research in another academic lab and get more training for very little pay, and that just wasn't something I was interested in at this time. So I went back to forensics, and I worked at the medical examiner's office for 7 years, doing pathology research and DNA methods research, how to make forensic DNA analysis better and more sensitive for mass disaster responses. So I worked on things that would help in an emergency. We used to simulate, different sorts of disasters that can occur. And a lot of people I worked with went to places like Thailand to help recover from the tsunami. The World Trade Center was a big part of that. Any sort of aviation accident was a big part of that. But I found my favorite research over the summer with me, and I would have, my trail of interns following me, and we'd train and we'd do poster presentations, and it was always my favorite part of the job. So when you find a part of a job that you love, look for ways to do more of that. And I saw the posting for Kingsborough. I applied here, and they were nice enough to give me the job. I've been here for over 10 years now. I love it. My favorite part of the day is still working with students and hearing how you interpret what I'm telling you, and hearing about your desired career path. So yeah, that's why I didn't stay in government or I'm still kind of I'm a city worker. But yeah, that's my career path. Norma is a very good world, in a different place now, but the fundamentals remain the same. Be open to learning new things. Never say no to someone who wants to teach something. Never think it'll come in handy. I got my first job in forensics because I was an undergraduate like you. I was sitting in my genetic engineering class senior year wondering what the heck am I going to do with a biology degree. It's very hard to find. It's not a terminal degree. That's in biology. You can do a lot of other things. And someone came in as a speaker from the New York State Police talking about forensic science and forensic biology. And this was before CSI was a show, so no one knew what he was really talking about. They're like, oh, that sounds kind of neat and weird, but no one was really talking about DNA yet. This was 27 years ago. So after the talk, he said, oh, does anyone need a job? And I was like, me. And I went up to him and he gave me his card, and I shadowed at the state police just to see what it was like. I had no idea what this forensic science was, and the people there were very nice and taught me what they were doing. And then later in the summer, a job posting came up, and I and several of my classmates actually applied for it. Many of us ended up working there as lab technicians. And then I had a mentor who was nice enough to take me under her wing for research. And she's like, you should really go get a PhD. And I was like, okay. So I did. That was it. But yeah, find mentors, see where they take you, and find what you love. There's a lot you can do with a biology degree, just maybe not right away. Medicine, research, science, industry, marketing, science communication is going to be huge for your generation. You need people to be able to talk about science stuff with people.
But yeah, I think now there's a lot of peer pressure when all of the undergrad have a lot of experience in like internships and in the workplace. Like it's just so competitive.
It is competitive, and it was that then too. As far as internships go, I did have one internship. My academic advisor was like, hey, there's this internship you should apply for. I ended up working at a Department of Energy lab as a research fellow when I was between my sophomore and junior year, and I got to live in Chicago for the summer, which was really cool. Yeah, and I was doing computer coding, which was not what I was trying to do in a biology lab, but because I used computer coding a little bit for my one semester of computer science class that I took, because I was under the impression I was either going to be a biologist or a computer scientist or a mechanical engineer, and I chose biology because I liked the class. But yeah, I did do that internship, and then the next summer I did an internship teaching junior high school students in Boston, Cambridge, Massachusetts, and that was a lot of fun. It was like a summer camp for students to learn. They did science, they did math, they did English, and I was one of the science teachers there. It was a lot of fun.
Thank you so much for sharing.
No problem. I like to hear your experiences too, while we have long incubation periods and things like that. But yeah, oh good, results are coming from, oh really? That's surprising. Let's see.
Is it really?
No, that's not. No, so sometimes it's easy to confuse. This is a hard test to read because it's so subtle. Can I see your positive, or did you get rid of it? It's a very subtle thing. It is not, see how there's like a pink layer on the top?
Yeah.
It's like.
Can I have a photo?
Yeah, sure. My unmanicured hand.
Thank you.
I don't get my nails done during the semester because they would get destroyed in the lab. see how there's like a pink layer on top? That's what the positive looks like.
I don't know why my phone keep having problem with the focus. Can you hold it?
Yeah,
Thank you.
it's hard to show it.
Yeah, it's very.
In summer internships and stuff, there's research opportunities here.
But yeah, it's
CRSP
clear.
program, and has a lot of research opportunities. There are a lot of hospitals here and medical centers and research centers that run summer undergraduate research experiences. And usually all you have to do is look at the school's webpage, like usually the medical schools or the other four-year colleges, they take students from all around the country, because I used to mentor those students as part of NYU's program and it doesn't hurt to apply. If that's something you're interested in doing, and it's a nice experience. They're very competitive.
Yeah.
But why not? Yeah.
I never actually been research at the same thing. I studied a little bit of studied some stuff, right? But I didn't see anything involved with biotechnology. So if you have a degree in biotechnology, what do you do for the military?
The military?
I'm supposed to go back on and go back and
I don't know, because I don't have a lot of experience. I do have
Yeah, I mean, it happens.
Yeah, USDA. A lot of things. So what you're learning now is definitely translatable to multiple areas. I mean, maybe not right now because the government funding situation.
Yes.
Area.
Stand for science.
Yeah, everyone's doing that. But yeah, there are. You never forget you're being trained as a scientist, and those skills, organizing, note taking, observing, finding patterns, are very good universal skills to have. Being able to communicate complex technical information, amazing skills. Don't forget to sell those skills when you go apply to internships, because those are things you pick up in the science majors too.
I read a lot of people talking about how so many students now graduate with biology degree but know nothing, and they kind of like upset with
Not if
the
they were in my class.
next generation.
Oh yeah, you got something?
Oh, yeah, this is
Yeah, that's
very
not.
good.
It's like, that's okay. It's not pink. Yeah, it's not pink. Okay, so let's look at our data and move on. Okay. So these are the expected results, so that's good. Enterobacterorogenies. So remember how we couldn't tell the difference between E. coli and Neorogenes based on the glucose, lactose, mannitol, the MR and the BP are a good way to do that. Because E. coli is positive for mixed acid, while Neorogenes is negative. BP is negative for E. coli, while Neorogenes is positive. So just a little aside, there's a set of tests that are usually used to distinguish among closely related bacteria. These are all gram-negative bacteria. So I'm not going to quiz you on that. It's just to know these are very closely related species. That's why we do these metabolic tests to tell them apart. And there's a set of metabolic tests that's usually used for these species specifically. You may hear the term around called IMVIC. So the IMVIC test, the little I is just for pronunciation, stands for indole, which we're going to set up today. That's a protein test. Methyl red, O's proscour,
Oh my God.
and citrate. So we've done most of the IMVIC tests at this point. We're going to look at citrate now. It's a really quick experiment. described as baking soda, which will raise the pH and turn the bromophenol blue from this emerald green color to this sharp sapphire blue. So what you're going to do is look at your tubes now. Like a sapphire, it's positive. If it's green like alcohol, or the leaves of a tree, it's negative. And if you're not sure, call me over.
I think my penis didn't grow.
That is one of my favorites.
It didn't grow.
That is
right?
negative. So here it's hard to say if it grew or didn't grow because it won't grow if it doesn't have citrate permease. Very striking test cell. Yes, that is definitely positive. And that does match theoretical as well, so that's good. Our screens are just being a little bit. We'll take it. What I'm going to do is I'm going to take pictures of this, so I have it, but I encourage you to do so as well, or take pictures of the chart just because that happens, because I do that. Warning label on anything. I give you the warning because I've done it. So now, any remaining tubes you have can go in the discard. We're done with carbohydrates for now. So we're going to set things up today the same way we did last week. Each row is going to get a different organism and you'll set up in pairs. I'm going to give you different organisms than you had the last time, just so no one gets bored. And we'll go from there. Now, we test that we go next week. I'll see you So, do that again. But remember, next time when we set up, you're going to need both weeks' lab manuals with you so you know what to do to set up your unknown tests. Okay, because that will be done independently. You'll have to know how many tubes to take, how to inoculate them, and what to do. You can use your lab manual. That's absolutely fine. I encourage it. But you're going to need to you're going to need to do it independently. So, I'll do this as we go. The four organisms remain the same. We have E. coli, E. aerogenes, P. vulgaris, and Pseudomonas aeruginosa. So, row one, you did E. aerogenes last time. This time you'll do Pseudomonas aeruginosa. What did you do, Bobby? You did E. coli, let's have you do P. vulgaris. You did Pseudomonas last time, great. So you'll do E. aerogenes. And you did vulgaris, so you'll do E. coli this time, and it works out perfectly. Everyone gets something new. So when I tell you to set up, I have 3 sets of the string, so you should have more than enough to set up between your pairs, because most of these are just a single sterile inoculation. There's no more duplicate tubes of these. So while one of your lab partners sets up, the other one watched, and you switch for the next test and let the other person inoculate. We'll keep it fair that way. Everyone should be getting experience setting these up, because next time you're going to do it on your own, so you do need these experiences. That's why you're here. So, gotta get used to it. So the first test we're going to go through is the indole test with the I in INVIC. The indole test is a test for deamination of tryptophan. So, we have this broth here. This is called tryptophan broth. It contains a lot of tryptophan, the substrate for the reaction that we're looking at. When you come up and get your tube, write TRY, tryptophan, whatever you want on the tube. But it will look like a lot of the other media next week. So make sure you label what it is, otherwise your experiments won't work. So how this works is we start with tryptophan in the broth, which I immediately spelled wrong. Tryptophan. And now I'm going to draw out the structure of tryptophan. I'm a little shy on this because I messed it up yesterday. So I want to make sure I don't do that again. So you have E double bond O, OH. Then you have carbon, NH2. That's your basic structure of an amino acid. You have the acid group, carboxylic acid. If you've taken OChem already, organic chemistry, you might recognize that whenever you see carbon double bonded to an oxygen with a hydroxyl group, that's an acid. Carbon binds to another carbon, which has an amino group on it, a nitrogen containing group. So amino acid. Attached to the carbon for every amino acid is what we call an R group. There are 20 different types of R groups. In tryptophan, you have a carbon attached to, and I'm very bad at drawing this, but I will do my best for you today. That's not doing well with it at all. They say organic chemistry teaches you to draw pentagons and hexagons really well. I took organic chemistry 27 years ago, so I'm not that good anymore. But it has this ring structure on here. And that ring structure is called an indole ring. I will not ask you to draw it. I just want you to see the parts because it helps this reaction make more sense. So, if the bacteria produces an enzyme called tryptophanase, it is going to deaminate tryptophan, which means it's just going to cut off the amine portion of the amino acid. So deaminate. And you're left with ammonia and the rest of this molecule. So further reactions cause this cleavage to happen. The indole ring comes off. So you're also left with indole. Does anyone recognize what this molecule is? 3-carbon carboxylic acid. It might look familiar from the reactions this week.
Carboxyl. Oh no.
What's a 3-carbon molecule that we looked at a lot this week? Starts with a P. It's pyruvate, yeah.
Pyruvate.
which goes on to the prep step of the Krebs cycle and is used in reactions further down the line to make ATP through oxidative phosphorylation. Or it can be reused in fermentation. So these are the three products you're making. Next week, we're going to detect if the bacteria made the enzyme tryptophanase by detecting this indole ring. When the indole ring is in solution, we're going to add something called Kovacs reagent.
Oh, what does it mean?
What was that?
Yeah, covert region.
Kovacs reagent. What is it?
I read on the lab menu, but I don't understand.
What do you mean you don't understand?
Like, what does it mean?
So we're going to throw our bacteria in the tube, and hopefully this reaction happens. Maybe it does, maybe it doesn't. Not all are positive for this enzyme. And what happens is I'm just looking in the lab manual, make sure I didn't miss anything to tell you. No, I got it all this time. So if the bacteria produces tryptophanase, it's going to make all these things and release them into the media. The indole ring is going to be detected by adding about 10 drops of something called Kovacs reagent. Kovacs reagent is like an oily yellow substance. So you're going to put a few drops on it. And if the bacteria, if the Kovacs reagent remains yellow, and a yellow ring at the top, it's negative for tryptophanase. But if you add the Kovacs reagent and it turns pink, it's actually like a dark red. It's like a dark pink. It's not as subtle as the Wilkes Pro scour, don't worry. Then it's positive for the enzyme tryptophanase. So all you're going to do this week is inoculate a tube of tryptone broth. We're going to let it grow over the week. And then we are going to add Kovacs reagent the next time we meet. For all these experiments today, leaving them in the drawer for 2 weeks is too long. It's going to make the experiments not work as well. And since it's spring break, we're not going to be here next Thursday. So what I'm going to ask you to do is I've set up a rack on this tray. It's right over here. I want you to leave all your tubes and plates today on this tray in this one rack. So make sure you label your tubes really well with the organism name, your initials, and the date so you can get them back next time. So organisms, initials, name of the test, date on each tube, the glass part of the tube. You're going to put it in here. I'm going to put it in the 37-degree incubator. Our lab technician is going to take everything out tomorrow so it doesn't over-incubate for us. Then, when we set up the protein test for the unknown, we have to set them up the same exact way we ran our controls, so we'll do this again for the protein controls when you set up your unknown. Just so everything's consistent. But for today, make sure you put your tubes here or they'll miss the bus to get incubated. Whenever you're ready, it's just an aseptic transfer into tryptone broth. If you need a refresher on how to do an aseptic transfer, we're going to be doing multiple ones today. Yep, it's right over there. The screens are right there. You can label it tryptone or TRY. I usually label this TRY for tryptophan broth. What is that? Why are you getting nervous? No, the antibiotic isolations we're keeping in our drawer for like 4 weeks. So you still need those. Please don't throw those out. I hope no one else did. So have things stay a long time. Yeah, you keep them. They're gonna turn like green and red and fuzzy. They look great. They're gonna smell even worse. Put it in a wrap, well labeled. It's not just enough now. Hang on to the strings because you'll be using them throughout the day today. And then we'll collect them at the end. The next test we're going to look at is phenylalanine deaminase. Just hang on to it because we're going to use it again and again and again. That way it saves you a little steps. Nothing in a microorganism is produced without a reason and a time for it. All these genes, as we'll see next week after spring break, in lecture review, we talk about gene regulation and genetic transfer in bacteria. All of these genes are very tightly controlled and very regulated, so they're only produced when the bacteria absolutely needs them. So by putting it at 37 degrees Celsius, that is the optimal temperature for these genes to be expressed. It helps trigger them. Okay, erase the board. We'll talk about phenylalanine deaminase. So that's found on the next page. All done with the chromosome. I'm going to just move it out of the way so we don't set up, and then hopefully we have more by the end. So one group might not set up this one pair. You might set it up as a row instead. Oh, you grabbed it. Okay, then we have enough. Thank you. Are we gonna have more phenylalanine though? The phenylalanine? Oh, your name. Okay, so at the end of class, I'll use it for my controls. Thank you. Okay, so the set we had was all contaminated, so we weren't going to use it. Okay, so phenylalanine deaminase. Like the enzyme name says, it's a deamination reaction, just like what we saw. So we'll draw phenylalanine, which is a little easier to draw than indole, thankfully. You have your acid group. You have your amino group. In phenylalanine, the R group is just a simple benzene ring. Just like that. So that's phenylalanine. Oops, I did draw it wrong. There's another carbon in between here. Very important to draw all the carbons. So you start with phenylalanine, and this time it's in slant form. So it's a slant. Remember when you inoculate a slant, you can hold it horizontally and just tickle your loop on the surface of the agar. This agar contains phenylalanine, which is the substrate for this reaction. So for phenylalanine, you inoculate your bacteria onto the phenylalanine slant. If the bacteria produces the enzyme phenylalanine deaminase, and you'll sometimes see this abbreviated PAD, which I welcome you to use when you write it on your tube. It's a lot shorter than phenylalanine deaminase. When phenylalanine deaminase is present, it's going to break down the phenylalanine into ammonia because it's deaminating, and something called, so we agreed last time the 3-carbon and acid was pyruvate. This is called phenylpyruvic acid because it still has the phenylalanine side chain, and then it's phenylpyruvic acid. So the other product here is phenylpyruvic acid. And this molecule can be used for biosynthetic reactions, not necessarily energy production. We are going to detect the presence of phenylpyruvic acid next week by adding ferric chloride, FeCl3, to the phenylalanine slant. We're going to add about 1 milliliter of ferric chloride. If the ferric chloride and the slant turn like a green, like kind of like Statue of Liberty green, like that rusty kind of copper color, we are going to call that positive for phenylalanine deaminase because the phenylpyruvic acid reacts with the ferric chloride to turn green. If the ferric chloride stays brownish, kind of a brownish rusty color, that's negative for phenylalanine deaminase, meaning phenylpyruvic acid was not present. So for this one, all you're going to do is inoculate a single phenylalanine slant per pair. And again, put it over in the wrap to incubate. I'm going to ask that you not leave spaces in between yours and your classmates' tubes because we're going to use them as space. If not 2 spaces. That will also keep the tests together so they don't get mixed up. So when you put your phenylalanine sensor, just put it in the next slot. Any questions on this one? Okay, have fun.
Now the meeting?
P80, okay.
You're just putting the bacteria on the surface of the slant.
And I put it back there, right?
Yeah.
Together. Because our hand body is different. We're gonna affect.
Oh, I'm sorry.
Okay. Ready? Doing other tubes over there. Other rack. Back. Yeah. Come over here. Okay. So the next test we're going to look at is the urease test. And that is the next one in your book. And all these chemical reactions are in your lab manual. All of them are also on the review slides, and there are also videos on each one in the lab review section. So if you need a little extra support on this, it's there for you. You can also ask me whatever questions you need to, either today as we go along or over email. Okay. So, my ear is bleeding. The urease test. So the urease test is a test for for detoxifying urea. Urea is a common metabolic waste product after protein metabolism. So urea,
Is it urine?
what? What was that?
Is it urine?
It's related to urine, sure.
Oh.
We make urea.
Oh yeah, because ammonia.
What?
Get ammonia, right? Because it's gonna break down.
It's going to break down into ammonia, yes. So urea, the structure of urea.
Can raise the pH.
So that's the structure of urea. And yes, breakdown products are very clearly going to be ammonia, which is part of this reaction. So, urea, if it builds up in the cell, and if it builds up in us, it's toxic. That's why we excrete it through urine as well as our sweat. That's why bacteria need to detoxify it and excrete it as well. So this is a test on whether the bacteria is capable of detoxifying urea using the enzyme urease. Urease is going to break down urea into carbon dioxide, water, and ammonia. See that right? Yes, we did. We have tubes here of urea broth. You're going to take one tube per pair and write urea on it. Notice it has an orangey red color to it. What pH indicator do we come to associate with that orangey red color?
Acid.
Not acid. What pH
Oh,
indicator?
pH.
Phenol red.
Phenol.
Yeah. So phenol red at neutral pH is this orangey red. So it's not acid this.
Yeah.
What does ammonia do to
Oh.
the pH of something?
Basic.
Makes it more basic,
Yeah.
right? It raises the pH. So we know that phenol red is red at pH 7. At pH, what does it turn yellow? 6.8, right? Super sensitive. You look at it a little funny and it turns yellow. It's very sensitive. But now we're going to go in the other direction. So, at pH 7.4 and above, also very sensitive. We're talking about tiny fluctuations in pH. It's going to turn this hot, bright pink color, otherwise known as magenta, as the pH rises. So if next week you don't add anything to this tube, next week you come in, if it's no longer orangey red, and instead it's hot pink, it is positive for urease. If it's red, orange, at pH 7 still it's going to be negative for urease. So again, all you're going to do is take a tube, aseptic transfer, leave it in the rack. And I'm just looking to see if any of my classes from yesterday's have turned already, so I can show you what that hot pink color looks like. So when you're ready, go on and take a tube. too. I appreciate it. Of course. You need the correct information, by all means, ask. Yesterday, I accidentally made 5 bonds to carbon, and a student corrected me, which I more than appreciate, because I can't have you leaving this lab thinking there are 5 bonds to carbon. Now we're going to talk about desulfurate.
Desulfurate.
We're going to desulfurate some stuff.
And produce gas.
So for the desulfurate test, we are going to use something called Kligler's iron agar.
Kligler.
Iron agar. Kligler's iron agar, as the name implies, contains iron. Also contains agar. It's also this orangey red color. Where's that orangey red color coming from? Phenol red. Yeah. It's our favorite pH indicator. So it also has phenol red in it. Has iron. And it also has glucose. Now, if you look this test up for desulfurase online, or even in our videos, sometimes you'll hear this test being done with something called triple sugar iron agar. Just know it's the same exact thing, but instead of just glucose, like Kligler's iron agar has, it has glucose, mannitol, and lactose in it as well. So triple sugar, 3 sugars, iron agar. We don't use it, it's more expensive, but also we already did it on tests. So triple sugar iron agar is usually used if you want to do all 4 tests at once. We don't, because we want to individually see which sugars are fermented and not, so we do the fermentation test. So what we're going to do with this is we are testing for a reaction called the desulfurase reaction. I am going to demo how to inoculate this media because it's different than what you've done previously. It's a double inoculation for the first time this semester. You get to use the needle. know, it's so exciting. It gets very monotonous, but I do want you to take a step back and appreciate just how good you all have gotten at aseptic transfers. Remember when you first came in here and you were like, I'm never going to be able to get this weird hand positioning, I'm not going to be able to do this, or you were like all shaky. And now you've just done how many aseptic transfers without me demoing them once, so great job. You're getting good, that's great. So, the desulfurase reaction is where we take amino acids that contain the sulfur groups. So, there are 2 amino acids that have sulfur in their R groups. So, phenylalanine with a benzene ring as an R group, tryptophan has an indole ring as their R group. Cysteine and methionine have sulfhydryl groups, which is just sulfur. Okay, sulfur. So the enzyme desulfurase can take the sulfur off of the R groups. And when it does, it produces, among other things, hydrogen sulfide gas. In the Kligler's iron agar, that hydrogen sulfide gas is going to react with the iron in the media and produce ferric sulfate, which makes a black precipitate. So, if you see a black precipitate form in your agar next week, that is positive for the enzyme desulfurase. If there is no black precipitate, then it's negative for desulfurase. And I actually have one to show you because I was looking at my stuff earlier this morning. I was like, one changed already, so you're in luck. So, not to spoil too much for you, this is Proteus vulgaris, and this is something I inoculated yesterday. You can see very clearly that there's a black precipitate in the tube. That is the reaction between the hydrogen sulfide and the iron that's in the medium. You may also notice that the tubes, some of them have started to turn yellow. Who can tell me what's causing the phenol red to turn yellow? Acid, right? Fermentation of glucose. However, for this test, we don't care. We've already done that test. We're just looking for the black precipitate. So, black precipitate, no black precipitate. You may also notice that that black precipitate is formed quite deep in the agar. How did my bacteria get there? I used the needle. Yeah. So this is usually produced best anaerobically. So we need to introduce the bacteria into the interior of the agar plug in order to get it to its optimal environment to produce desulfurase. Put that back, let it keep cooking. So the way to do this inoculation is what I like to call a double inoculation. You stab first, and then you spread on the surface. So I'm going to inoculate one desulfurase tube to show you. So just like we do always, I'm going to have both my tubes in one hand. I have my culture tube here. You can come around if you need to see. That's fine. Just don't block anyone's out. Start with the needle. Like we always do, 10 seconds in the sterilizer. Let it cool. I'm going to do an aseptic transfer first of my bacteria from my culture tube into the agar plug, just by dipping the needle into the bacteria and then dipping it once into the agar. Once or twice is fine. What I don't want you to do is mash up the agar too much because that'll introduce air deeper into the agar, and we don't want that because we want an anaerobic environment for this. Contamination. then just like a sewing machine, I'm gonna go straight in, straight out, straight in, straight out, and that's it. Sterilize my loop again, my needle again. Then once it's sterile, I can put it down, cap up my tubes. Now the second part of the inoculation is with the loop, just on the surface of the slant. This is shallower slant than you might be used to, but the principle remains the same. You sterilize your loop and just tickle the bacteria onto the surface of the agar with very light pressure. Cap it up, put it in the rack over there. Remember to label the tube KIA for Kligler's iron agar along with your organism name and the initials. Because again, this might not look the same next week. Any questions? You can watch me inoculate the slant as well if you need a refresher on that. Fine. Well, the inside of the tube is sterile, so I'm just contaminating the tube with the bacteria I'm putting in anyway, so it's okay if you touch the inside of the tube. Touching the outside of the tube is because that has environmental microbes that we want to avoid. But the inside of the tube, I have shaky hands. If it were a problem, I would never be able to do it.
everywhere.
Everything okay?
Yeah.
Time for us to get stuff from the play?
A lot of stuff.
Yeah.
Exciting.
So, it's still. Yeah. It's like mold.
So your plates from the antibiotic that you did with the dirt dilutions last time, they still need a few more weeks to grow. So what we're gonna do is we're gonna look to see if anything orange, green, like looking rusty brown color comes up, because those are usually the ones that are gonna be producing the antibiotics and the pigments. So they still need some time to grow. They are gonna smell really bad, so open your door after surgery, it's gonna smell like very earthy garden smell. All right. Two more steps to go, 3 more tests. The next test we'll discuss is the nitrate test. And this one, I wish more people hadn't stepped out for a moment because this one's complicated. The nitrate reduction test of your lab or manual right before bioremediation. So, what do we call it when a bacteria uses a final electron acceptor in respiration other than oxygen? Anaerobic what? Respiration, right? Anaerobic respiration. So the nitrate reduction test is a test for anaerobic respiration using nitrate. And there are lots of different types of anaerobic respiration. Some use sulfate, some use carbonate. We're testing specifically for anaerobic respiration using nitrate.
Nitrate.
So to do this, the filters out of the way, we are going to use something called nitrate broth. As you can imagine, nitrate broth looks just like all the other broths, except it contains a lot of nitrate in it. So make sure you label this to NIT, nitrate, whatever you want. The nitrate reduction test we're going to do to test to see if the bacteria can use nitrate as the final electron acceptor in anaerobic respiration. So we start off with nitrate. That is nitrate.
And nitrite.
Don't get ahead of me. Nitrate is an oxidized form. It has 3 oxygens. Okay? This becomes important. I want you all to remember the mnemonic right now that I showed you. Leo says verb. Loss of electrons is oxidation, gain of electrons is reduction. This becomes really important in this test. So nitrate can undergo decomposition into NO2, which is nitrite.
Or nitrogen.
Notice the only difference between those 2 words is an I. Nitrate
Trait.
gets reduced to nitrite. There are fewer oxygens in the nitrite. It's reduced. Every time it goes down a step, it is gaining an electron. Okay. Getting reduced, gains an electron. Nitrite can be further reduced to nitrogen gas, which is the most reduced form. When this happens, we call this denitrification. This is the most reduced form. Which means it's holding all the electrons it can possibly hold. Its hands are full if it had little atom hands. Hands are full. Can't hold any more electrons. It's the most reduced. This is the process by which soil bacteria return nitrogen to the atmosphere as part of the nitrogen cycle. Nitrogen can then be reassimilated into living things through the process of ammonification, where the nitrogen combines with hydrogen to become ammonia. This is done by soil bacteria called rhizobium that live in the root nodules of leguminous plants, which we're going to dissect later this semester. I just started growing peas in my backyard with a little bit of rhizobium starter. You can get it at any garden shop. It's usually called inoculant or pea booster. Unfortunately, we've not had the best weather, so I'm hoping I don't have to replant them, but if I do, I will. But we're going to dissect those root nodules later in the semester so we can see the rhizobial bacteria, which are responsible for ammonification, otherwise known as nitrogen fixation. The bacteria takes the nitrogen gas from the air, converts it to ammonia. That ammonia can then be used to make proteins and nucleic acids by living things. That's how the nitrogen cycle continues. We'll talk all about this in weeks 9 and 10 when we talk about environmental microbiology, microbial ecology. But for now, nitrate is the most oxidized, nitrogen gas is the most produced. There are two enzymes that catalyze this process. The first is nitratase. That's going to be responsible for the breakdown of nitrate into nitrite. Then you have nitrite going to nitrogen gas by nitritase. I'm always going to emphasize that I, because again, the words are very important. Nitritase takes the nitrate, nitrite, and converts it to nitrogen gas. So in this test, the reason a lot of students find this test so complicated is, the first reason are, you're not looking for a positive or a negative. There are three possibilities here. A bacteria that is capable of anaerobic respiration using nitrate. First possibility is it only has nitratase. So it only takes the process this far. Still capable of using nitrate as a final electron acceptor. It's just not capable of denitrification. So that's possibility one. Possibility two is it has both enzymes. And is capable of denitrification. The third possibility is it has neither enzyme. And is not capable of anaerobic respiration using nitrate as a final electron. So there are three possibilities here in one test. So when you mark your results next week, you're not only going to say positive or negative, you're going to say positive or negative for what enzymes. This could be positive for nitratase only, positive for both nitratase and nitritase, or negative for both enzymes. So the way we test this is a two-step process. The first part of the process is we're going to add to the tube next week nitrate A and nitrate B. These are two reagents. These names I commonly forget, and I'm not going to quiz you on it either, but it should be sulfonilic acid. Yep, nitrate A is sulfonilic acid, nitrate B is alpha-naphthalene. Don't worry about it. Nitrate A, nitrate B. When you add nitrate A and nitrate B, if nitrite is present, it's going to turn red. That means it's positive for nitratase only. And you stop. You're done with your test. That's possibility 1. However, it's a possibility that there will be no color change. If after you add nitrate A and B, there is no color change, you have to keep going. Because it's still a possibility, either 2 or 3 is the case. Okay? You need to distinguish between both enzymes being present or neither enzyme being present. So, if both enzymes are present, nitrate is going to be completely reduced to nitrogen gas. It's gaining and gaining electrons till it can hold no more. What we're going to do to test to see if nitrogen gas is present, or if neither enzyme is produced, nitrate will still be sitting in that tube next week. The bacteria will have done nothing to the nitrate. It's still there, which is the more oxidized form. Plenty of room for electrons. So that's what we're distinguishing between. To distinguish between these two possibilities, we're going to add zinc powder. Zinc is a strong reducer. It has a lot of electrons to give. It wants to give electrons to everything in sight. However, if nitrogen gas is present in the media, the nitrogen gas is going to say, so what, I'm already holding up, I can't take anymore. Don't give me any more electrons. If nitrogen gas is present, and there's no color change when you add zinc, then it's positive for both enzymes. So this is the weirdest situation where no color change is a positive result. Because it's already holding all the electrons it can hold. The zinc isn't going to make a difference because everything in that tube is already reduced if both enzymes are present. However, if you add the zinc powder and the media turns red, that means it's negative for both enzymes. Negative for both enzymes. That's because the zinc gets into the tube, sees the nitrate, and says, oh, you can hold more electrons, and gives it all to them. And then the zinc turns red. That's why that works that way. So it's not only a matter of positive or negative, you're going to have to make an observation. When does it turn red? After nitrate A and B, or after zinc, or not at all? And then that's positive for what enzyme is negative for what enzyme. The string is in your book as well. Just drew it out the same way. And it's also in the videos and the lectures. So all you have to do now is label one of these culture tubes, nitrate, your organism name, your initial disease, sterile aseptic transfer. We're running out of room in the rack.
I don't want to wear this anymore.
Make sure you put the date as well. It is. This one's a complicated one. If you take Bio 51, this is the last thing we do in lab. up today, I want to go over the bioremediation report because there are parts you can start before we collect data. Just so if you want to start talking about that with your group, you can. But we won't be setting up those experiments till after break.
That unclear.
Just talked about a task for anaerobic respiration. Now we're going to talk about two tasks. One is for aerotolerance, or the ability to survive in the presence of oxygen. The other is for aerobic respiration itself. The reason I'm going over these tests together, aside from the fact that they're related by oxygen, is the fact that they're set up on the same medium, which is a TSA plate. So if you're not quite done with colony purification just yet, do it at least a couple more times. So the first test we're going to talk about is the catalase test. And you were a little bit when we talked about oxygen and growth requirements. So if a bacteria can survive in the presence of oxygen, it needs to be able to detoxify the byproducts of aerobic respiration, which include superoxide anions, which are very toxic to the cell. So one of the byproducts of aerobic respiration is the superoxide anion. It is super reactive. It's a free radical. It likes to bind with everything. It likes to alter the functions of different enzymes by binding to them. It's very toxic to the cell. But if you're going to survive in oxygen, you're going to encounter that. So you need an enzyme to detoxify it. And that's an enzyme called SOD, or superoxide dismutase, which takes this very toxic superoxide anion and converts it into hydrogen peroxide, which is only a little bit toxic to the cells. However, if a bacteria is going to be aerotolerant, or tolerant of oxygen, this includes fabricated anaerobes, aerobes. Basically, the only ones that don't have these are obligate anaerobes, or something that is extremely anaerobic. So, you need to be able to detoxify hydrogen peroxide, and you do this through an enzyme called catalase. Catalase takes toxic hydrogen peroxide and converts it to completely non-toxic water and oxygen gas, which we observe as bubbles. So, the way this test gets set up is each pair is going to take one TSA plate, label it with your organism name, the date, your initials, and you'll label this one oxidase and catalase on the agar side. Next week, on a colony, you're going to take some hydrogen peroxide, and you're just going to put a drop of hydrogen peroxide on a single colony or area of growth. And you're going to look to see if bubbles rise up. If there are bubbles, it's positive for catalase. If it's negative, no bubbles, which means it is not aerotolerant. not have the enzyme catalase, which helps detoxify the byproducts of aerobic respiration. The oxidase test is a little bit different. The oxidase test is also found in your book. The catalase test is not. Catalase test, oh, it is. We put it in. Way to go us. We just converted this to an accessible lab manual, so I'm still checking and hoping everything made it in. If you notice something missing, please let me know. The catalase, the oxidase test, where are you? What? Right behind catalase, thank you. Oh, yep, there it is. We're using the last place we want. Okay. So the oxidase test is a test for aerobic respiration. After all those wonderful electron carriers like NADH and FADH2 are reduced, they need to hand their electrons off to something. Eventually in aerobic respiration, what does it get handed off to? Who's the final electron acceptor? Oxygen. But in the meantime, it goes through some electron carriers as part of the electron transport chain. NADH passes its electrons off to the first electron carrier that's embedded in the bacterial cell membrane. Proton gets kicked out. The electron travels to the next electron carrier. Another proton gets kicked out, and you make a proton gradient across the bacterial cell membrane. This also happens in our mitochondrial cell membrane. In aerobic respiration, this continues until the electron is passed off from something to oxygen. In aerobes, that electron carrier is called cytochrome C.
That's
And
the
it
gene I
is the
use in the
electron
research.
carrier that's responsible for passing that final electron off to oxygen to make water. And lots of ATP through that chemical gradient. So, cytochrome C in its reduced form, holding the electron, undergoes a reaction with the enzyme cytochrome C oxidase. And what results is the oxygen receiving its electrons and making water, because that's the final product of oxidation of glucose, water, and carbon dioxide. So as the electron, and E apostrophe is just shorthand for electron, is passed to oxygen. And oxidized cytochrome C. This is loss of electron to oxygen, so loss of electrons is oxidation. We are going to try and detect this oxidized cytochrome C, because it's only going to be oxidized if aerobic respiration is taking place, by adding something to a colony on the TSA plate that you're making. That's why it's important to do a quadrant or a 3-street to get single colony. You're going to add the oxidase reagent. And I only say the name of this chemical twice per semester. It's tetramethyl P phenylene diamine dihydrochloride. You can call it the oxidase reagent. I am not going to quiz you on any of these actual names. It's just important to know them in case you read a label that isn't labeled oxidase reagent, that you know what it is. But for now, if you add the oxidase reagent and it turns black, that means it's positive for cytochrome C oxidase and thus is capable of aerobic respiration using cytochrome C as one of its electron carriers. If it stays pink or colorless, depending on the brand of reagent we buy that year, It is negative. Now you have to be very careful with that negative. Because the negative doesn't necessarily mean it's not capable of aerobic respiration. It just doesn't conduct aerobic respiration using cytochrome C. There are other electron carriers. However, if it's positive, you know it's capable of aerobic respiration using cytochrome C. So to do this, all you're going to do is do a colony purification onto a TSA plate. If you need a review of it, I'm going to wash the chalk dust off my hands and do one up front. If you feel confident and ready to go, you are more than welcome to do so.
Look.
I just need to get my hands all undusted.
Oh, look, my eczema.
So we're doing one for Kaylee.
Yeah, you can get it for free.
No, you're only going to take one plate.
One plate.
And you're going to use that plate. Just a little drip on one side, a little drip on the other. Yeah, you don't need 2 plates, just one. So to start my streak, I'm going to sterilize it. Let it.
So we do it on one half of the plate.
So you're streaking a whole plate today. But next week, you're going to add 1 chemical on one side, 1 on the other. The streak remains the same as we previously done. So I take my first loop of bacteria, do my first streak, and I'm doing the pre-streak.
So just swap it or we do 4?
You do the quadrant streak. So you do want a colony purification. So I'm going to take this, go over my top streak.
I'm still not very good at that.
And I sterilize it.
Get more practice.
Catalase and oxidase. I label my plate OxyCat. Because you're going to use it for oxidase and catalase. And you're going to put it over on there. So for my second streak, I'm going to make sure my drip is cool enough first. If you're a sizzle, it's not cool enough. Then I'm going to take some bacteria from my first streak and pull it down a few times. And do my second streak across. Just back and forth, back and forth. Then I sterilize my loop again. Now I'm going to do my third streak, which is my last streak using this technique. I'm going to let it for about 30 seconds. I can make sure it's cool. Then go the rest of the way down. When you're done with this, or if your partner is doing it, you can clean up your bench and make sure I get the culture tubes back. Correct. Just hold tight, because then I'll hand out the liquids for the bioremediation. tax